That being said, there may be a few of you who want to get out in the sun immediately right after the clicker questions, so let's get those done and over with as quickly as possible. So the reason that the rhabdovirus capsid has a different shape than paramyxoviruses is the GFMPCV or L protein. Ten, 5, okay, we're done. What do people think? Nobody likes PC or V or L. That's good because those are the polymerase proteins, have nothing to do with the capsid protein. Nobody thinks the F protein has anything to do with rhabdo versus paramyxos. Why? Do rhabdos have an F protein? No. Okay, so we're down to G and or M. So what's the G protein? The receptor binding protein, the fusion protein, particularly true for VSV. Does that have anything to do with the shape? It's the matrix protein, which forms a helix in the capsid, which makes that difference. So our next question. Stuttering by paramyxovirus RDRP is important for host range, avoiding host defenses, 3' poly A tail formation on mRNAs, 3' poly A tail formation on the genome, or B and C. Ready? Ten, 9, 8, 7, 6, 5. Okay, so to give you a bit of an idea here, this actually should have been C and D, but it's still wrong. So it's not such a big deal. 3' poly A tail formation on the genome. Can't be that. Why not? Because these are negative strand RNA viruses. So you don't want to have a poly A tail on the genome. It would be totally worthless. So poly A tail formation on messenger RNAs. Yeah, I like that. Avoiding host defenses? Host defenses? What the heck? Did we talk about host defenses when we talked about paramyxoviruses? Very, very little. Turns out, however, that for the virus we're going to be talking about today, the filoviruses, it may actually be really important for that. So, but not for the paramyxoviruses. And the host range, that's going to be whatever your functional proteins are going to be, either for binding on the outside or potentially whatever you need to replicate on the inside. And you could say that stuttering is important for changing the open reading frame for PC and V. What I didn't tell you about is that that may be important actually for host range, but we didn't talk about that in class, so that's fine. So yes, it is C, the poly-A tail formation on the messenger RNAs.
I have a question.
Yeah, sure.
So in the book, it was saying that haltiness, or the frame shift caused by stuttering on the P gene causes the presence of V and W, which help in avoiding host defenses.
Yeah, so these are, again, we didn't talk about it in class per se.
Okay.
And so, but this is, again, I meant this to be C and D
Okay.
rather than that. And so it's may well be critical for that, avoiding the host defenses. And again, important versus critical. These should probably be better written questions. And so given that, we'll probably, I'll have to go back and check and give people credit for both of these. That work?
Yes.
Yes? Good? Happy? So this is why if there's like a question on the exam that you're like, what the heck? Send me an email. And it may be something, and that's why I also post keys immediately after exams, so you can take a look at them. Thank you. So about what is the death rate in known cases of measles? 0.1, 1, 5, 10, or 50? Again, these are the questions that I love to write because coming up with possible answers is easy.
Which does not give you autism.
10.
Doesn't give you autism. Screw the scientists.
So yes, it has nothing to do with autism. I completely agree. Oh yeah, that paper's been retracted and it's a mess. So almost at 100% here. Very close. So if anything, it's probably lower than 0.1% in terms of these cases, but certainly very small percentages. But even that being said, with small percentages, you have to be careful. And so the hundreds of cases that we have of measles in any given year in the U.S., because people aren't vaccinating for the most part, is still going to be causing some deaths. On the other hand, 50% down here is the mortality rate for the filoviruses that we're going to be talking about today. So very different in terms of numbers here. Enjoy the sun. Yeah. Pardon? Oh, so for filovirus, there are no decent filovirus vaccines currently. So that's probably why. we hide this. Sure, yeah. Question.
So are you just posting the clicker scores at the end of the term?
Oh yeah, so the question is about clicker scores. When are you going to see them? When I get around to posting them. But basically, at this point, everybody except people I sent emails to who took the midterm, I have your clicker scores, and I'll post them. But there's still a few people who haven't let me know which their clicker is to put the two together. So that's why I haven't posted them yet.
So we should be seeing clicker scores on D2L?
You should be not yet, but when I post them, yes, you will. But you should not be seeing them there yet. No. And plus, I've got to do stuff like correct today's clicker questions, which will take some time. Other questions, comments, worries at this point, other than there's way too many photons outside. Well, it's beside the point, at least for today's class.
Aren't you going to Berkeley?
Actually, the weather's worse there.
Really?
Currently today, yeah. Colder, cloudier. So, yeah, why do I bother? If you're interested afterwards, I'll tell you why I'm going. So, any more questions about these Paramyxo and Rhabdoviruses? Again, the important aspects here really have to do with replication and how you're making your messenger RNAs with this whole start-stop mechanism in terms of how the polymerase is going through, and then, of course, the structure of the genome being single-stranded, negative, single genome viruses. Filoviruses, they're basically really big rhabdoviruses in terms of their molecular virology. The reason that they get their own chapter, of course, is this hemorrhagic fever aspect of the filoviruses. But basically, molecularly, they're pretty much these big rhabdoviruses. Again, big, long rhabdoviruses. RNA editing takes place in these guys, again, very similarly to what happens in terms of the paramyxos and rhabdos. Same kind of stuttering aspect. And again, it makes sense because that's what the RDRP is doing anyway in terms of making poly-A tails on all the messenger RNAs. And then, of course, the hemorrhagic fever aspects of things. I was going to bring my cute little stuffed model of Ebola, but didn't do that. Anybody know giant microbes? Yes. Yeah, so the Ebola apparently is one of their best-selling of the giant microbes. And this is the electron micrograph, sort of the classic electron micrograph of Ebola virus. You can, this should be F.A. Murphy, not F greater than A Murphy. If you think about it, what kind of biosecurity conditions do you want to be in to be taking an electron micrograph of a virus like this? Just something to think about. So yeah, we'll talk a little bit about the structure, the genome, the proteins, editing, regulation, and then flipping stuff around a little bit. I'm going to talk about disease at the end because I've got a couple of videos and we'll see how well that ends up working out as far as class time goes today. So the structure basically looks a lot like a big rhabdovirus. But again, the sort of take-home message here is that we've got enveloped, single-stranded, negative genome. In the envelope, glycoproteins sticking in the outside. What do? Interact with receptors and then cause entry. You have, unfortunately, different names for everything in here, but basically your matrix protein is the VP40 protein, and again, it's that matrix which is holding this long, flexible structure together. And VP24 is another membrane protein. I'm not going to expect you to remember all these names because there are way too many VPs. But there is one that is really quite different relative to the other viruses which we've talked about so far, and that's this VP30, which is a transcriptional activator. And we haven't really talked much about transcriptional activators, so we'll take a little bit more closer look at VP30 a little bit later on. Nucleoprotein, NP, L protein for replication. Basically, again, just like in the Paramyxos and Rhabdos, pretty much everything the virus needs to function is in the virion, so on the outside of the cell. If you look at the genome, it's bigger than most of the Paramyxo and Rhabdoviruses, so about 19 kb, so large size, but otherwise extremely similar in terms of its organization. Got a leader sequence at the 3' end, a trailer sequence at the 5' end. These are identical sequences to each other. People argue about whether the rule of 6 is true in these filoviruses, but certainly it appears that those sequences at the ends are really, really important. So you have to have the exact sequence at the end, both 5' and 3'. Again, that's probably having to do with replication, may or may not have anything to do with packaging. You've got the same kinds of start and stop intergenic sequences, these gray bars, which you have up here at the top, where you have a short stretch of U residues, which will then get copied over into giving you poly A tails by pretty regular stuttering. The only sort of major difference in terms of the genome organization here is that some of these genes overlap with each other in terms of their start and stop codons, whereas in the Paramyxo and Rhabdos, which we have down here at the bottom just for comparison, none of them seem to actually do that. Whether it makes a difference in terms of how they're being regulated or not is not entirely clear. Yeah?
I don't know if you said this, but how big is Rhabdovirus
It depends on the actual rhabdovirus. Rabies, if I remember correctly, is about 17,000, but it's not that different. Just that they're a little bit smaller in terms of the genome. Certainly in terms of the virion itself, they're much shorter, but that's because the M protein is making this helix. So they're shorter and wider as opposed to longer and thinner. So yeah, these, again, overlapping open reading frames, but they're clearly very different relative to each other. So let's see. Yeah, other things. So the other aspects of this, again, mentioned before, pretty much all these other proteins are the same. The big differences are this, particularly the VP30 protein. I would ignore VP24 for now. Strong activating effect on viral messenger RNAs. And so it's a very specific transcriptional activator. We'll see how it's functioning a little bit later on. Curiously, it's present in Ebola, but not in Marburg. And these things give extremely similar kinds of disease phenotypes. So it's probably not that critical for disease, but important for replication of the virus. And that's one of the things that researchers are trying to figure out is if you're trying to look at disease and trying to come up with therapies for any of these filovirus diseases, you try and find things that are going to be different. But the fact that you've got something here that it's probably going to be really important, right? Activating transcription, but you don't need it in this other one that's giving you, again, very, very similar kinds of phenotypes. So interesting from a molecular biology point of view, probably not that important for a pathogenesis point of view. But I like the molecular stuff, so we'll talk about it. How do these guys get in? It's the GP protein. Again, they've all just got different names relative to each other. Basically, almost identical in terms of how they function. All slightly different, however, in terms of how they're put together. So the GP protein, this is practically identical between Marburg and Ebola. Made as a precursor protein. It gets chopped up usually by the furin peptidase, which we talked about when? Previously? Furin peptidases? Cellular protease is important for maturation of viruses, so it's got to be an envelope virus, right? So yeah, it's the Flaviviruses, which are also using furins in terms of getting cleavage to take place in your fusion peptide, your receptor binding protein. Turns out you can get rid of the furin site, and again, the virus is perfectly happy in terms of replication. So is it required? Is it not required? Probably it's a pretty flexible structure in any case. This is what it ends up looking like cartoon-wise. Transmembrane helix, cleaved usually by furin, but it doesn't seem to be absolutely necessary. And also contains the fusion peptide. hidden inside the structure. So what happens once it gets inside endosomes? Conformational change, fusion peptides exposed, inserts into the host membrane, you get fusion that takes place. This protein is extremely highly glycosylated, i.e. lots of sugar residues attached on the outside. That's why it's called the glycoprotein, just because of all these sugar residues that are attached on the outside. Probably that helps it escape the immune system. So a lot of these sugar proteins on the outside is often very important for immune system escape. Did anybody make it to the biology seminar yesterday? You talked about modifications to glycoproteins, and that seems to be, particularly for innate immunity, what those sugars are is really important. Again, it probably happens in the ER and the Golgi. Golgi is where you find furin, where you're getting this binding trimer. What the receptor is still not entirely clear. People are trying to work on this. One of the big problems, of course, with working with these filoviruses is they cause really nasty diseases. And so working on them is much more challenging than working on a lot of these other viruses. So the sort of classic techniques that you use to find receptors are much more challenging to do in some of these cases. So we don't know yet. And that's also true for some of the viruses. It's easier to work with as well. How do they get put together? Again, basically they're Paramyxo and Rhabdoviruses. Start-stop. In terms of when you're making your messenger RNAs, as soon as you make enough of your nucleocapsid protein, that coats the genome, and so you go from making negative strand to positive strand. That also is enough of the nucleocapsid protein that then binds, makes your genome sequence. And so, again, extremely similar to what's happening in the Paramyxo and Rhabdoviruses. It's a little different than some of those is that you have budding that happens at the plasma membrane, as opposed to budding which is happening in Golgi's or the pre-plasma membrane kinds of lyses. One of the major differences between these filoviruses and the Paramyxo and Rhabdoviruses has to do with what's happening with editing. So again, this, probably in these cases, again, textbook says that we've got PCV, which is causing host, or actually the Y, it was a Y and M that's different. Yeah. Right. So those actually, I'll maybe have to go back and check that, because those are different start codons rather than RNA editing. So I may have been right in that clicker question. I better go back and check. But here, instead of causing changes that, again, between PC and V, which are probably important for replication, here stuttering's happening in the GP protein. And so what happens there is you end up with a different C-terminus when you get stuttering that takes place. And so these are just the cases that you have in the genome. It looks like here, up at the top, you end up with the SGP, which is the secreted glycoprotein, which is probably really important for host defenses. In this case, it's pretty clear that that's what's going on. On the other hand, if you have stuttering that takes place, now you have addition and frame shifting of one adenine residue, which gives you now a membrane-bound protein. So you secrete a bunch of this glycoprotein, which is, if you don't have stuttering that takes place in this open reading frame, if you have stuttering, then you make a membrane-bound form. So, and people have done this now, where you mutate these sites. So if you look at this run of A's, again, stuttering, where you're going to get multiple nucleotides that are the same. Sometimes the RDRP is going to add extra nucleotides. If you now change some of these nucleotides, in this case down at the bottom, to guanines instead of adenines, these are still coding for lysine because of the wobble position at the third position of the genetic code. You can end up with just glycoprotein as opposed to glycoprotein and secreted glycoprotein. And in that case, what you end up with is something which seems to not cause as much disease in terms of the host defenses. Now, of course, in this process, you've also shifted and given yourself the frame shift. And this has all been done through recombinant DNA and particular mutagenesis. Probably, again, if this were to happen in a naturally occurring isolate, it wouldn't spread through the population as well, wouldn't be causing as much disease. So, but this, I think, is a really nice indication of how you can use molecular biology to figure out what's going on here, and that this slippage here really is due to this whole run or homopolymer, as you say, of, or as we say, of these nucleotides. So that's one of the, again, big differences between the filoviruses, or small differences, most of them are pretty the same, between filoviruses and Paramyxos and Rhabdos. The other one is what's going on with VP30, which is, again, specific to Ebola, not present in Marburg. If you look at the NP, also known as N protein, this, again, is what's going to be producing the nucleocapsid protein, which is what you need to package your genome. It's the very first gene that you have present next to the leader sequence, so at the 3' end of your genome. It has a very strong secondary structure, this hairpin structure that forms at the beginning of that open reading frame. If you have VP30, that basically removes this block to transcription, and you get a lot more. This hairpin blocking transcription is interesting because it's a transcriptional regulator, but it's a function which forms in the RNA. And probably what's happening here is you're getting base pairing that takes place. Because remember, your messenger RNA is going to be base pairing with your genome. And so probably what happens is this forms a stable structure on your genome and is blocking making more of the genome. And if you remember, again, that's right at the 3' end. And so it's going to be blocking all the transcription that happens through the rest of the genome as well. And probably the reason for this, again, to totally over-anthropomorphize, is that you have a buildup of your virus. You get enough transcription, and this doesn't block all of it. If it blocked all of it, you'd never even see, well, you've got the VP30 protein in there anyway. You have an infection, and the VP30 protein, which is in the virion, can overcome this problem. So if it's just your positive strand RNA virus, you had structures like these at the beginning of the genome, you'd never see any of your proteins. But since VP30 is together in your virion, that will allow this to take place. But it probably also means you have to have quite a lot of that VP30, which comes in, maybe even multiple infections that take place, which may be why actually coming down with one of these diseases is relatively difficult, which we'll talk about in just a second. A lot of these experiments have been done with recombinant filoviruses. This is just an image on how that's done. We talked about already for the positive strand RNA viruses, it's relatively easy to make an infectious clone because it's an RNA. That RNA can be translated, make the proteins, etc. Negative strand RNAs are a lot harder to make. And the trick that was done here in terms of being able to put together these viruses in a recombinant fashion, making those point mutations to look at secreted GP versus GP, that was done by our friend T7 RNA polymerase. The problem is at the 3' end, and so the 3' end, what they did was they engineered in a ribozyme. In this case, it was the hammerhead ribozyme, which cleaves itself. So basically, it makes an RNA, and then the last little piece chops itself off. And so you end up with exactly the right sequence at the leader and trailer sequences, so the 5' end and at the 3' end. The RNA, of course, by itself can't do anything. You have to assemble all of the proteins on that RNA in order to get it to be functional. And so you've got to express all of the various different proteins, usually on individual plasmids. To do this, you do want to be in a relatively biosafe, secure environment. Anybody know what this is or where this is? Galveston, Texas, UT Medical Branch. This is the BSL-4 lab, which is, I think, fascinating place. I actually did go through here, but all BSL-4 labs are inside BSL-3 labs. So it's a lab inside a lab, a building inside a building. And you can kind of see right here in the middle, were you not getting any windows? It's It's right behind there where the BSL-4 facility happens to be. They all have these offsets. Nobody can drive any closer here. Everything is blocked off, and it is airport-style security to be getting into even the building on the outside of the BSL-4 facility. I thought it was interesting that this BSL-4 facility was built right in the path of hurricanes. It's like, why would you do something like that? And at first I was like, these guys are nuts. But I later actually talked to some of the architects who were doing this, and they said they prefer hurricanes to tornadoes and earthquakes because you can see them coming. And they designed this building in such a way that it was supposed to be fine in terms of hurricanes. And they did have a hurricane. I forget which one it was. It came through Galveston and Houston a couple of years ago. The BSL-4 lab shut down completely during the hurricane. 2 weeks later, it was up and running. So you also notice it's up a floor. So any storm surge, everything is coming through down here. My poor collaborator who's in Galveston, his microscopes were totally trashed because they were in the basement, got completely flooded. But the BSL-4 lab was fine. So it does seem a little odd to be putting a BSL-4 lab right in the middle of a hurricane zone, but if you design for it, apparently it works okay. So that's it for molecular stuff. Now we'll tell the cool, gross stories about these viral diseases. So the first of these filovirus diseases, strangely enough, was identified in this beautiful small German town called Marburg, university town. This is the castle up at the top of the hill, where it was first identified in the 1960s. Again, right in the middle of Germany, but of course that's not where these filovirus diseases come from. Eventually, they figured out it was importation of monkeys from Africa, which had led to these disease cases. So most of these, this is the image from the previous version of the textbook, but it's also very similar. If you look at Ebola virus, about 500 cases so far, at least in terms of the last edition. It's now gone up to about 800 cases. Almost all of these in equatorial Africa. The original African outbreak, if I remember correctly, was here in 1976. Relatively small areas, and again, very much in equatorial Africa where these have happened. And what's interesting is you see outbreaks of the disease, so again in 76, and then nothing for a few years, and then another outbreak and nothing, another outbreak and nothing, which tells you a little bit about the spread of the disease. It seems to be pretty self-limiting. These don't spread very far, and as many people I've talked to say, they're pretty wimpy viruses. They have trouble actually getting from one host to another host. So we had all these cases in Africa, and then Reston, Virginia? Where does that come from? So turns out that this is a place quite close to Washington, D.C., where a lot of animal work is done, particularly with people from the NIH, Bethesda right here. And a lot of the monkeys were imported usually from Asia, not from Africa. And it turns out that they were coming down with disease that looked a lot like the same kind of hemorrhagic fevers that people were getting from these filovirus diseases which were found in Africa. Fortunately, none of those seem to transfer to humans. So the monkeys get really sick, but their handlers don't. And people have, in fact, found that they probably have been exposed, the handlers, because you can find antibodies to these viruses, which turns out to be really nice because now you can actually work with this form of Ebola virus, not having to do absolutely everything in BSL-4 because it doesn't seem to be causing disease in humans. Be very careful about your monkey colonies, but you don't have to worry so much about working with that. Now, of course, people are very interested in what are the differences now between these two different strains. And is that a way that will tell us something about the pathogenesis of some of these filoviruses that you find in, in Africa? Why do we care? Well, very high fatality rates. And so that's the big deal. Depending on the outbreak, anywhere from 30 to 90. Some of the earlier ones were 90% fatality rates. Now we're getting closer to about 50%. Hemorrhagic fever, basically you bleed out of all of your orifices, which is kind of ugly. So that is why people also certainly notice this. The infection, sort of the host, seems to mostly be macrophages. So blood cells, and that kind of makes sense for excess bleeding. Where do these viruses hang out? What's their reservoir? Probably these cute little fruit bats up here, in which they seem to cause no disease whatsoever. But again, as we talked about before, how do you know if a bat is sick? But they're certainly not bleeding out of all their orifices. So and I would not want to do these kinds of experiments. So go into the bat cave where you think these guys are infected with nasty human viruses and collect samples. A lot of times they're talking about we're talking about the rectal sample of a bat. It's not something I want to think too much about. That being said, even with looking at those samples, it turns out it's still very difficult to get infected by one of these viruses, even in the presence. And so probably that aspect of going and infecting the bats, looking at the bats, the virus numbers there are very, very low. And so the risk is actually not that high. And we know this because if you look at disease, it's almost always very close family and medical personnel who end up getting infected by these diseases. And most of the time with medical personnel, it's a needle stick. It's something where people have just not been as careful as they really should have been, which is a challenge, particularly if you think about where a lot of this is happening. It's really very much developing world kinds of diseases. Clearly immunosuppressive, that probably has to do with the infection of the macrophages. Why do we care about it? Probably because of a lot of the media. And there are probably about 3,000 total cases now of both Marburg and of Ebola. Some of you may not have seen this. All of our friends going out in their BSL-3 gear. There's one thing that's really completely missing in here. Where's their air supply? We can go and talk about that a whole bunch more later on. The update that I had last year, 2012, I guess 2 years ago now, there was a case in Uganda, and then it was in May, and then 7 cases and 4 deaths, 24 cases, 17 fatal, 77 deaths. But the numbers here are minuscule in terms of what's going on here. A lot of people are concerned about Marburg. In 2012, there were 15 cases, 8 of those probable of those 15 deaths. 1 tourist died in 2008. So think about Marburg, people being worried about traveling into these countries. 1, as far as we know. Tourist has died from Marburg fever. A U.S. tourist was infected and survived. There's a whole branch of the CDC looking at these things. Why? You probably heard that there's now currently an Ebola outbreak. Interestingly enough, this is not in Central Africa, but more in West Africa. 234 cases so far, 62% fatality. Unfortunately, still a certain number of healthcare workers who have been infected and also have died from this disease. There is, and I didn't bring this up, but if you like, you can actually go and download exactly how to deal with Ebola and people who are infected, etc. This is an example here, in fact, of a CDC scientist working in, I believe this is Congo, a previous case where they were there. The basic message that this thing says, if I remember correctly, I think it's barrier nursing. So basically gloves, gowns, etc. Really want to avoid any kind of blood transfer, or also just bodily fluids in general. Where is this happening? Again, it's in West Africa. Liberia, Guinea, maybe in Mali. As you could see, most of this is in the bush, well away from most of the concentrated centers, except for a number of cases here in the capital of Guinea. That's because the people were brought there to be treated. The main treatment basically for all of these viruses, because there aren't vaccines at this point, there's no decent therapy, is basically just to isolate people and then treat the symptoms, which I could imagine as a healthcare professional, I wouldn't be real happy with something that's a 50% plus, 63% plus percent fatality rate, to saying, okay, here you are, I'm going to shut you off from everybody, and good luck. Which is basically, unfortunately, what the current therapies are as far as dealing with these situations. So just to finish up here, wanted to mention the ideas that how we think these diseases, particularly Ebola, but it's true for many of the other zoonotic diseases, are being spread. Most of the time, these viruses are circulating in bats. This is from the CDC, by the way. So I should block that out. Circulating in bats. Occasionally, these bats will infect non-human primates. And interestingly enough, this antelope seems to be a really susceptible to Ebola-like disease and has all of those nasty symptoms. So if you see a lot of dead antelope around, you want to be pretty careful and probably not be eating these dead antelope. Chimps are also a possibility. So usually these are getting infected and then somehow this gets into humans, pretty much always very close family. Again, because of the blood, etc. One of the problems is symptoms first take a while to show up, and second, sometimes look like other diseases. And so those original symptoms. And so that's potentially why you have very often primary caregivers or even pre-primary caregivers are going to be family. And they at that point don't know. So education is also something which the CDC and various health agencies are trying to deal with. What are we trying to do in terms of dealing with this? Vaccines, particularly subunit vaccines, so expressing the GP protein on a completely different virus. In fact, VSV is one that they're using a lot to try and get an immune response, which then could be used, hopefully, in terms of vaccines. One of the nice things about the fact that the disease takes a while to kick in, you can probably do post-exposure vaccination. Certainly in animal models, this has worked quite well. So just like rabies. In rabies, you can vaccinate after exposure. And in this case, hopefully you can vaccinate after exposure as well. A lot of these, again, are through recombinant viruses. So you've taken one of the proteins, and again, the GP is the really obvious one to use, and put it into another virus. Some people are talking about using RNAi. So small interfering RNA, usually hairpins. These will be double-stranded RNAs that match some part of the virus genome, which then you've got double-stranded RNA, and then we've got a whole system of dealing with double-stranded RNA in the cell. Yeah?
Are they looking into inhibiting VP30 as well?
Yeah, so at first people were all gung-ho about inhibiting VP30, but since it's not present in Marburg, they're like, okay, it might help with Ebola, but it's not going to help that much with Marburg, and so people are trying to find something that's going to be more general between Marburg and Ebola. So they were really excited about it to start with, and then probably not going to be that good as a therapeutic. Okay, so that's all I had in terms of the molecular biology, the disease aspects. I have a couple of videos that I could show you, but it's a really nice day outside, and it's Friday. So what do you think? Sayonara. Have a good weekend. This one, by the way, at the top here, just to mention really briefly, this is from This Week in Virology, where they took a camera crew through the new BSL-4 lab in Boston before it actually went online. So it's really pretty unique footage about how these labs are put together. So I think it's really nice, but it's way longer than we have for the rest of today.